Hello everyone, I'm Tong Wang from Microsoft Research. Today I'm happy to share our research on AI2BMD, from static protein structure prediction to dynamic characterization. AI2BMD redefines protein dynamics with initial accuracy. As we all know, all bioactivities are dynamic. For example, the photosynthesis, the respiration, the nitrogen fixation, and the enzyme catalysis. Recently, the Nobel Prize in Chemistry 2024 was awarded for protein structure prediction and protein design. Although the static protein structure prediction has achieved great success, the accurately characterizing the dynamics of the proteins is much more challenging and need to be addressed. As we all know, the molecular dynamic simulation is a widely used way to study the protein dynamics. MD simulation is to simulate the atom's movement in a protein or other biomolecular system in the real environment by approximations of non-physics. So it can be seen as a virtual microscope with a high temporal and spatial resolution. And in the MD simulation, one of the key components is the force field. It drives the movement of the atoms and updates the atoms' coordinates. The MD simulations can be roughly divided into two classes. The first is classical MD simulation, which employs Newton's laws to drive the atom's movement. It can perform long-time simulations for exploring the conformational changes. However, the accuracy of classical MD simulation is not very high. On the other hand, another kind of MD simulation is quantum simulation, which uses quantum mechanics. It has high precision for describing the electron transitions. However, due to the high expensive computational cost, it cannot be used for biomolecules or proteins. So here we propose the AI-powered MD simulation, and it may become the third way to study the protein dynamics with a balance between the accuracy and the efficiency. The organisms consist of many cells. In each cell, there are many proteins that plays key roles in regulation. And in each protein, there are many atoms. Here, we use deep learning and artificial intelligence to calculate the atomic force for each atom and moves its positions accordingly. Therefore, the molecule has been moved by the AI-driven force field, and by the long-time simulations, we can detect the biomechanisms and design drugs. Our AI2BMD's paper, Initial Characterization of Protein Dynamics with AI2BMD, has been published online on Nature. Here is a brief introduction of the technical pipeline of AI2BMD. First, the proteins was fragmented into several protein units. Then, we generate the data sets for the protein units at the density functional theory level. And then, based on the dataset, we train the VisNet model as the AI2BMD force field. By the AI2BMD, we calculate the energy and atomic forces for each atom, and then simulate the protein dynamics. With the simulation trajectories, we can get the kinetics and thermodynamics properties, and then compare with a wet lab experiment. We analyze the calculation accuracy of AI2BMD. Here, we select several proteins for the evaluation. We use the mean absolute error of the potential energy and atomic force as the evaluation matrix. Here, we use the results calculated by DFT as the reference value, and we compared AI2BMD with molecular mechanics. As shown in the figure, the red bar shows the error between AI2BMD and the ground truth, and the green bar shows the results calculated by molecular mechanics, the classical MD, and the ground truth. It's very obvious that AI2BMD significantly reduced the error compared to molecular mechanics for the potential energy. As for the atomic forces, we can get the similar results. AI2BMD achieves significantly smaller errors than molecular mechanics. We then evaluate the calculation efficiency for AI2BMD. As shown in this figure, you can see we compared AI2BMD with density functional theory, a widely used approach of quantum simulation. With the increase of atom numbers, AI2BMD has achieved less time consumption than DFT. We then compared AI2BMD with other machine learning force field and AI-driven simulation system program. And from this table, you can see AI2BMD costs less time than other AI-driven simulation software. We then simulate the protein folding and unfolding processes by AI2BMD. As shown in this figure, starting from the unfolded structure of signaling, AI2BMD can fold signaling with 10 nanosecond simulation steps. And during this simulation, we can see the errors of AI2BMD is smaller than the errors made by molecular mechanics. As seen in another figure, you can see the unfolding process. From the folded structure, AI2BMD can unfold the signaling structure. And also, in this simulation trajectory, you can see the error of the potential energy made by AI2BMD is significantly smaller than that made by molecular mechanics. Then we made thermodynamics property calculations by AI2BMD. Here we choose two protons, Barnese and Ci2, for simulations at the different temperatures. Then we use AI2BMD for the calculation. We analyze the enthalpy during the folding process. As shown in this figure, the enthalpy calculated by AI2BMD is much closer to the experimental results, which is much better than the molecular mechanics. And then for the heat capacity calculation, the results made by AI2BMD is also close to the experimental values, which is much closer than the molecular mechanics. Then we analyze the folding free energy during the simulation. As shown in the figure, the curves made by AI2BMD is well aligned with the experimental values, while the has a gap with the experimental values. This result shows that AI2BMD can accurately calculate the thermodynamics properties. We use AI2BMD for drug discovery and drug virtual screening. We took part in the first global AI drug development competition. In this competition, more than 1,000 teams around the world took part in this competition. There are two tasks, which are both to predict whether a small molecule can bind to the main proteins of SARS-CoV-2 and whether it has the inhibition ability. We use AI2BMD for the structure modeling, the simulation, and analysis, and achieved the best accuracy for both tasks. And finally, we won the championship during the competition. In the future, we will use AI2BMD for the important biomechanism detection, the understanding of important bioactivities, and we will use AI2BMD for the drug discovery, the biomaterials design. We believe AI2BMD can open more opportunities for detecting the biomechanism and design new drugs, and provide more potential values for life sciences and precision medicine. For more details about AI2BMD, please refer to our paper published on Nature and the code repo on GitHub. Thank you.